number 2. Both of them bind, but one binds more than the other one. Let's say ligand 1 binds more. So it binds more compared to this one. Delta G. Both of them binds, that means delta G for both of them is going to be positive or negative. Both of them bind, so it's going to be negative because they are spontaneously binding. Which one is going to be binding more? 1. So negative value is going to be more for which one? This one or this one? This one. So delta G for this one is going to be both of them negative, but this is going to be more negative. Let's say, for example, this one is negative 9 kilocalories. This one is negative 7 kilocalories. Got it, guys? Everyone got it? Negative, because they are spontaneously binding. Both of them are binding, but one of them binds more. So it is going to have more binding affinity. Okay? So when we discover a drug, let's say you're making 2 or let's say you are making 5 molecules. Molecules can be ligand, okay? Ligand, okay? Whatever it is. L1, L2, L3, L4, L5. In the lab you did it, and you have a protein that you expressed. Like you have a protein that you want to know if these ligands bind, if they bind or not. Okay? So you have 5 of them, and you found L1. This is a different example, okay? A different example I'm showing, not this one. Let's say L1 binds more than L2, but the highest binding is coming from L4. This is the highest binding. How do you know that? By calculating the binding free energy. If this one has the largest, let's say this one is negative 10 kilocalories, this one is negative 5, this one is negative 7, this one is negative 8, this one is negative 6. So which one is going to have more binding affinity? Which one is going to have more binding affinity, guys? Ligand 4, right? Ligand 4 will have the binding more. So this is how you are working in a biochemistry or biology lab, and if you are trying to make a ligand, whether it's going to be useful for our purpose, you always want to see which one binds more. Okay? So let's say COVID. You know Paxlovid, right? We discussed that before. The main ingredient of Paxlovid is what? It's called nirmatrelvir. Some of you have done that, right? In our presentation. Anyways, nirmatrelvir made by Pfizer, right? Pfizer made thousands of molecules in their lab, okay? And then they found that nirmatrelvir binds more with the main protease in our body, okay? That's how they say, oh, this might be a good candidate for therapeutic purpose. Do you understand what I'm saying? Once you understand, it's gonna get easier. All right? So, here we are talking with respect to free energy, delta G, change in free energy, correct? But the problem is, experimentally, we cannot measure free energy change. We can measure something else. That gives you free energy, indirectly. We measure dissociation constant. What? Dissociation constant. This equilibrium constant. Okay, so this thing. Or equilibrium constant, or equilibrium binding constant. Sometimes they also say that. So equilibrium constant, to calculate that. Or so we can measure that. So how do they measure it? They can take the concentration of this and of the complex, divide by the concentration of the ligand and protein, and then they get the K equilibrium. Okay? That K equilibrium, then they convert it to free energy by using this equation. What is this equation? It is called, it is delta G equals negative KB T ln of K equilibrium. This one, not this one actually. This one. This is the equilibrium constant. Okay? KB is the Boltzmann constant. B stands for Boltzmann. This is the constant value. Okay, Boltzmann constant. Do you know what is Boltzmann constant, guys? Have you heard of gas constant? What is gas constant? In gas law, in GenChem, and in many other fields it is used. It's 8 point, do you remember 8 point something? 8.314 kilojoules per mole per degree Celsius, something like that. That's called gas constant. That's for 1 mole. If you divide by the number, Avogadro's number, you get Boltzmann constant. So what is Boltzmann constant? Boltzmann constant is Kb equals R over N. N is the Avogadro's number, and R is the gas constant. Gas constant divided by the Avogadro's number. Both of them are constant, right? Gas constant is a constant value. It never changes. Avogadro's number is a constant value. What is the Avogadro's number, guys? We discussed that. What is the Avogadro's number? Starts with what number? 6, right? 6.023 times 10^23, right? That many number makes Avogadro's number. So Avogadro's number, that means your gas constant divided by Avogadro's number gives you another constant. Both of them are constant value. If you are dividing constant value with the constant value, you are going to get another constant value. Never going to change. Let's say the constant values are 5 and here you have 2. That's going to give 2.5, right? So this value will never change if they don't change. Correct? That's called Boltzmann constant. So Boltzmann constant times T stands for the temperature in Kelvin. Ln of K equilibrium. K is the unknown value. You can get it from experiment. So you can get the K value from experiment. These are the constant values. You can calculate the delta G. This T is the Kelvin temperature. Let's say you are doing the experiment at 30 degrees Celsius. What is the Kelvin temperature? 30 plus 273, correct? So it's going to be 303, correct? So this T value depends on the temperature of the lab. The lab that you are using, if they are using air conditioning, maybe it is 20 degrees Celsius, then you have to add 273 with 20, that's going to become 293 then. Okay? Kb is a constant value. So Kb constant, T you get from the thermometer, and you measure it from the experiment, you measure the K equilibrium. You multiply this, use the ln, you are going to get delta G. For binding, you are going to get this value. Got it, guys? So, experiment you get this one, K equilibrium. So, now come here, K equilibrium. Okay? So, if you know K equilibrium, you can calculate the delta G using delta G. Let me write it here. Negative KB T ln of K equilibrium constant. Alright, guys? This is what you need. Alright. Now, if you look at the I have some class activity in case you need it. I still have some. Okay, so one of the question is remember, you are not going to get all the questions from activity. Please make sure you also read the lecture note. Okay? Okay, so if you have, let me show you a slide that's gonna show you something. Okay. Okay. You see the formula here, right? This is the equation, right? What do you learn from here? It says that delta G value is going to be negatively large when K equilibrium value is low. Okay? If the K equilibrium value is low, delta G is going to be large. Let me explain. All of you wrote this part. Now we are going to write something a little bit differently. And again, I'm going to give you my example. If delta G value is negative, but a very large value, let's say it is 8 kilocalories, del K value is going to be larger, and we convert it to micromolar, okay? 1.5, let's say. But if it is delta G is a smaller, you see -5 I'm writing. This one is larger, this one is smaller. I mean, don't concentrate on the negative value, because negative might mean that it is smaller than this one, right? But it is actually negative, means it's actually going down. That means it is forming the complex mode, okay? So just consider that 8 has the highest binding affinity. Negative just means that it is spontaneously going to make it, okay? More spontaneous. Now the K for this one, because this is a smaller, you see, this is larger, negative large. So this is larger. This is gonna be maybe this is 0.7 micromolar. You understand what I'm saying? Okay? So no, I think I'm going on the opposite. 2.0 micromolar. This one is a smaller, this one is larger. You see? This one is a smaller, this one is larger, right? This one is larger, this one is a smaller. So always K value, if it is large, that means I'm making a mistake. Just a second. Let me explain with another example. Don't want to say something wrong, okay? So better find it, and then from my own work I can explain it. It's always a confusing thing. Okay. Yeah, I should have kept that slide. You saw this slide in my lab, right? Now we're going to show this one. Look at this. We measured the KD values This is the dissociation constant, equilibrium dissociation constant or equilibrium constant, 16 nanomolar, and this is the 1.8 nanomolar. And we showed that this one has more binding. So Kd value is small means binding free energy is large. Did I say that? Yeah, that's what I wrote. Kd value larger, smaller, here the Kd value is smaller, means delta G value is large. Okay, that means the binding affinity is large. Binding higher in this case. Okay, so binding is higher. In this case, the binding is lower because delta G is smaller. Kd value of the dissociation equilibrium constant is larger. Got it, guys? So always remember that. Always remember that if the Kd value is or K equilibrium constant is lower, binding affinity is higher. Okay? Alright. Everybody understood what I'm saying? Yes.
So KB, KDQ, they're all equals to, they're all the same.
That's where I'm going. There are two things. Kd means dissociation constant. Okay? Kd means if you have a protein ligand complex, then protein ligand going to protein plus ligand. That's called protein ligand complex. I'm not going to go in that detail now. I'm just going to call you in the papers or in the research, if you are doing, if you see that Kd, that means the equilibrium dissociation constant is higher, that means it has a lower binding affinity. Okay? So concentrate on this thing, Kd, equilibrium dissociation constant. If the equilibrium dissociation constant is higher, binding affinity is lower. You understand what I'm saying? Just remember that. If the equilibrium dissociation constant is higher, binding affinity is lower. Okay? So remember this, if delta G is higher, that means equilibrium dissociation constant is lower. Got it, guys? So remember this, what I'm writing. Delta G is higher if Kd is lower. They go like oppositely. Okay, they are not same. They don't go like before. If delta G gets bigger, Kd gets bigger. It's not like that. If delta G goes higher, Kd goes lower. Remember that. Okay. So if I ask you a question, can I remove this, guys? Okay. If I ask you a question saying that you have two ligands, L1 and L2, or like that, Kd values are, Kd value is 1.2 micromolar and this one is 0.8 micromolar. What would be the delta G? Which one will have the higher delta G? This one or this one? Select from higher or right, higher, lower. Tell me which one is gonna have lower? Delta G. So this one will have lower delta G? Higher? Okay. This one is higher. This one will have lower. Everybody agree? So remember that. Again, I'm not going into detail. If you are really interested about KD, more, come to me. If I want to explain it, everybody will be confused now, okay? I don't want to go in that detail. Just go there, just try to understand that one. This one is lower, that means it has a higher delta G value. That's what you remember, okay? For now. Okay. All right, so let's go back. So overall purpose is to know if we are developing a molecule that is going to be useful or not. That's what is our purpose here in this class, okay? Now you know that this one is going to be useful for us, right? Because it has a higher binding affinity, delta G, all right? Because it has a lower 0.8 micromolar. So if you are measuring this KD in your lab, you always want to make sure you get a very lower KD value. Got it, guys? All right. Now you know that this molecule work, but this one also has a binding affinity. You now want to do another experiment, saying that, okay, how much doses do I need? How much concentration of these molecules do I have to use? So for that, we have to use something called IC50. What is IC50? Inhibitory concentration to reduce or to neutralize the function of the protein by 50%. Sorry guys? Again, I have posted all the lectures, activity, important things are there. Please make sure you do that. Do I have it here? I didn't bring it. But I have posted those lectures. Oh yeah, here it is. So what is IC50? IC50 refers to the concentration of a drug inhibitor to inhibit a biological process by 50%. Okay? Now look at this. These are two drugs. Let's say these are two drugs. Okay? This is one. I'm not going in detail. Let's say this is ligand number 1, this is ligand number 2. So let's rename them as ligand number 1. This is ligand number 2. Okay? What do you see in this picture? This is the neutralization, percent neutralization. What is neutralization, guys? Let's say a COVID virus goes inside our body. Remember replication? COVID virus increases in our body. You are using a molecule to stop this replication by 50%. Oh, sorry, stop this replication by 50% is here. 50% is here. Okay? That's called neutralization by 50%. At 50%, what is the doses or what is the concentration of the ligand you have used to neutralize by 50%? So this represents concentration of the ligands. Okay? Let's say you have, let me draw it, you have a test tube. You have your protein. Okay? Let's say this is a protein. That is used to replicate the virus in our body. You want to neutralize this protein so that it doesn't replicate the virus. So what you do? You add the ligand. first you add ligand very small amount, 0, then 0.1, 0.0001, 1 microgram per milliliter. That's the concentration you are using, unit. So you are adding very small amount. When you add a very small amount, it doesn't affect. It doesn't neutralize the protein. You keep continuing, you are adding, and when you reach here, you see now it is corresponding to 50% here, right? So 50% neutralization for ligand 2. What is the corresponding concentration? Corresponding concentration is here, right? What is it? Maybe 0. What? 0.02. No, 0.012. Okay? That's the concentration you need for ligand number 2 to achieve 50% inhibition. Very good? Everybody got? So you are adding, like we take the medicine, like the small drug, the pill that we take, right? You take it with a very small amount, and it is doing a lot of things in our body, right? We don't want to take like the food that we eat. We don't want drug molecules like we are eating bread or something like that. We take a very small amount because they have side effect. So we want to give a very small amount to reduce to neutralize the virus, right? So we are adding very small amount, and with L2, you see ligand number 2, you need only 0.012 microgram per milliliter concentration of ligand 2 to inhibit 50%. But to inhibit the same protein with L1, ligand number 1, you need more, right? So this one is about 0.018 microgram. So which one is more effective? The one that you added very small amount, right? It is neutralizing the virus. Okay? In this case, if you are thinking about virus, it is neutralizing virus. If you are thinking about a protein, it is inhibiting the protein's function by 50%. Okay? So this is what we look at when we discover a therapeutic drug. Okay? We first look at the KD value, and then we don't stop there. We say, okay, now we want to use it to see if it can inhibit the protein's function. If it can neutralize it. Neutralize means that the protein is not going to work anymore. Okay? So we look at 50% because it takes time to go to 100%. Sometimes it can take a long time. So we look at 50%. That's the standard. Okay? Standard is we look at the concentration that you need for the ligand to neutralize the protein's effectiveness by 50%. Okay? That's called neutralization. Everybody got it? So now if I say you have L1 and L2, now you have IC50. IC50 for one of them is 0.01 microgram per milliliter. So let's say this is L1, and now you have L2. Let's talk about L2 and L3. Maybe you get another ligand. And then you have this one, 0.018 microgram per milliliter. Which one neutralizes the protein? L3 or L2? L2, right? L3, right? Because it has higher, it has L2, not L3. L2 because it neutralizes with a very small quantity of the concentration of the ligand. Okay? So write it down, what I am writing now, and then think about it, because these are going to be coming in your exam. I'm going to be asking, not same, but I'm going to be asking relevant questions, because these are the most important things in biochemistry that you can learn. Okay? All right. Number 7, I just explained it from your activity. Number 6 is this one. Number 7, I already explained. Number 4. Number 4 is, there is a computational method that we normally use to calculate the binding free energy, because experimentally we cannot calculate binding free energy, but computationally you can. So how do we do that? We use molecular dynamics, and from that molecular dynamics we calculate the binding affinity using binding free energy using two things. Using a method called MMGBSA. Now look at the thing. There are many other methods that are used, but I'm just explaining one of them. This is the delta G of binding. Again, what is binding, guys? Protein ligand bind and make the complex, correct? That's called the binding. So ligand binding. How do you calculate the binding? There are computational methods. Experimentally we can calculate the KD, but we don't know the interaction energies. What are the contributing factors? But computationally you can calculate the binding free energy from VDW is the van der Waals interaction. That means all the non-bond interactions are there. Non-bond. Remember we discussed about that? Non-bond Lennard-Jones potential and all those things. These are the non-bond interaction. This is the electrostatic interaction. This is the polar solvation energy, and this is the non-polar solvation energy. And I have written them here. MMJBSA is known as molecular mechanics generalized Born surface area solvation. Okay, so this is what it is called. It is a method to calculate binding affinity between protein and ligand and protein complex. Okay, protein can also bind. Two proteins can also bind. So how do we calculate the delta G? Using van der Waals electrostatic interaction, delta G of the polar solvation energy and delta G of non-polar solvation energy. Okay. Now you might be wondering, what are those things? I don't understand. What is it? Right? Do you know the polar interaction? Polar molecules like polar, right? Everyone understand that? Like dissolves like, polar likes polar, right? Polar interactions are there. You see, polar interaction, non-polar interaction. When they are not non-polar likes non-polar. Let me explain you with a picture now. Can I remove this? We are done with this now. We have a protein. Let's say this is our protein. P stands for protein. Okay? Let's say this is our ligand. Ligand means a small molecule. I'm just showing the cartoon representation. This is a ligand, that means it's a molecule, okay? Molecule. Again, we already explained it, I'm again saying it to you. You add the two together, you get protein ligand complex. Protein ligand complex. Everyone got it? Clear? Okay. Now, in our body, do we only have protein and ligand? We have a lot of water in our body, right? Everywhere, even in the cell, in our skin, we have water. It looks like we have nothing in there, right? We still have water inside the skin. In the flesh, in muscle, everywhere is water. So they are actually surrounded by the waters, right? They're surrounded by the water, correct? And they have to remove these waters from here to make the water protein ligand complex, and then again make the water surrounding it. Correct? So these are the waters. we also have salt, like different ions, calcium ion, sodium ion, potassium ion, iron, many things, right? So we need to include them somehow. So how is it doing it? In this protein, when they interact with the ligand, there has to be some attraction between the polar, right? So these are the polar interactions. They come in the Van der Waals interaction term. All the polar, non-polar, between the ligand and protein. Ligand and protein, they have to interact. Okay? So Van der Waals, VDW means Van der Waals interaction has to be there. And it has to favor the attraction, attractive force. Then it's gonna make the complex. Another one is electrostatic. You know, in the proteins, we have carbon bound with carbon. Remember the protein, in protein, what do we have? Amino acids, correct? We have to define that structure. Protein and the ligand. Because let's say this is a chemical compound, a benzene. Let's say the ligand is benzene with a methane. So this is our ligand. You see, they are a covalent bond. These are defined, and in the case of protein, we have amino acids. You have to define the molecular interaction with this term and this term. These are the two. And also the polar solvation energy, the amino acids that are polar likes polar. Remember we said that polar likes polar. And non-polar. Also in the polar, we also have water interacting with the amino acids that are polar, right? We know polar molecules like water, right? Polar amino acids like polar. So all the contributions are coming here, and here. And if they favor the binding, if the delta G value, after calculating all these four, if the delta G value comes negative, then we can say that it favors binding. Got it, guys? So we are calculating the interactions for all the sorts of interactions that can exist between protein and ligand, and protein complex, protein ligand with water, protein complex with water. Got it, guys? Yes. What I'm saying? Not only water, ions also. If we include the ions in our calculation. All those things you combine, some might be negative value, some might be positive value, some might be negative. So if the total, let's say this one is 10, this one is 12, this one is 5, this one is 7, your total energy is going to come negative, right? Negative something. 10, negative 2, then it's going to come negative 10. Got it? So then you are going to get negative value, and that means our protein ligand will like to bind. Got it, guys? So these are very interesting if you focus. Just try to understand that, and then it's become easier for you. So now here we are showing, you see, this is our delta G binding. This is from a real calculation. This one is negative, this one is negative, these are positive, this is positive, this is negative. If you combine all of them, all four of them, it's gonna come negative 6. That means it favors binding. All of them favor binding, but this one favor more. So we can say that this one is gonna be our important ligand for binding. Okay? Do you understand what I'm saying? So try to look at these slides. I know it's not going to be easy, but from the slides, if you don't understand anything, ask me. Okay? Alright, so that's all. I think we are almost done, but here we are going to talk about a little bit. We are going to start virus, so this is where the virus starts from. And yeah, that's it. So in our next class, we are going to start virus. There will be a new slide, but we are going to start.